Everybody, we're back, and we just were talking about what a species is and different ways that we can define a species. And one of the things we ended on is that a species, um, in order to become a different species, at least in the biological species concept, will not be able to mate with one another. Okay, and so in order even for that species to develop into a different species, what we have to have there is first as a reproductive isolating mechanism, a way that restricts gene flow between the species. The most obvious one we think of as a physical barrier, and we're going to go over that, but they're not always physical barriers. There's a really nice table for you in your textbook, table 20-1. And so that lists all the different types of isolating barriers. We can broadly categorize them into two main groups. So if it's a prezygotic barrier, that means that the two different species or the two individuals are prevented from mating from one another at all. So no fertilization can actually take place, right? So that means no zygote can form. That's why it's called prezygotic barrier. Okay, so no zygote can form. That means no fertilization can take place. Maybe they go through a mating act, but the egg and the sperm can't fertilize. So you can't develop a zygote. So that's a prezygotic barrier. And we're going to go through 5 of those, I believe. If two individuals do mate, right, and a zygote forms, then that's called a postzygotic barrier. Okay, the zygote forms, but then the development gets halted or the success of that individual gets halted. Either they die or they can't produce offspring. That's a postzygotic barrier. Okay, so again, 2 broad categories: prezygotic barrier, postzygotic barrier. So let's look at the 5 prezygotic barriers. Okay, and here are the 5. So you need to know these terms, right? Nice vocabulary terms for you. Temporal isolation, habitat isolation, behavioral isolation, mechanical isolation, and gametic isolation. I like this picture here because she's kind of being a barrier for mating. He wants to mate with her and she's making him work for it anyway. She might prevent it. Just a silly example to show you a type of prezygotic barrier. Again, you're going to prevent the actual mating. Okay, so what is temporal isolation? Temporal, remember, means timing. Okay, so if it's temporal isolation, it means that the 2 species end up reproducing at different times of the day. It could be at different times of the year. It could be only certain seasons, right? And so here's a wood frog, right? And the leopard frog. The wood frog mates in the early spring, and it's usually before the ice has completely melted off of the pond. So that's still kind of cold, but they can tolerate that. And that's when they choose to look for mates, right? The leopard frog will usually not mate until a few weeks later. And here you can see on the chart, you can see mating activity as it goes up on the y-axis, and then time on the bottom. So time, again, is temporal isolation. So the peak mating activity for each of these is at different times of the year. And so they probably wouldn't encounter one another. Okay, there's a possibility that some individuals that go into mating early might encounter those individuals that mate late. But oftentimes when that happens, these are the less viable individuals. They may, for whatever reason, not be good partners. So they might even be rejected by the other. Okay, so temporal isolation. Different times of the day, season, or year. I mean, it can even be like time of day. Some insects mate in the daytime, some insects mate in the nighttime. So they just don't encounter one another. This is often hormonally driven, okay, with temporal isolation. The hormones to drive the mating behaviors peak, and they might get stimulated by photoperiod, right? And that can happen again, between day and night. But photoperiod also changes over the seasons. Remember when we looked at reproduction and you looked at external reproduction and you had videos of coral reefs releasing egg and sperm into the water, right? That gets triggered by the moon, by a full moon. So there's a lunar cycle that triggers mating with those coral. Other coral might use a different signal, okay? So temporal isolation, if they're not being triggered at the same time for their mating situation, then they'll never, there's no gene flow, right? That's a pre-zygotic barrier. There's no gene flow there. You can click on this from Canvas, and cicadas are a good example of this. Cicadas, their offspring are buried in the ground, and they develop underground, and the adults only emerge. It depends on the cicada, like every 17 years or something like that. So their timing is very important for their mating. Okay, so temporal isolation, timing. The timing of it means only gene flow can occur during that time. So if you mate at completely different times, there's no gene flow. Pre-zygotic barrier. Next one is habitat isolation. So habitat isolation, again, pretty obvious here, it requires different habitats, right? And so it's the requirement of their habitat that promotes their mating behaviors. Okay, so here's a least flycatcher compared to an Acadian flycatcher. The least flycatcher mates in the open woodlands. They need the openness of the woodlands. You know, they may be relying more on sight to find one another rather than sound. Whereas in the deciduous forest, very lush forest, harder to see one another, but they might be able to hear each other. So they might use sound in order to attract one another. Okay, so with the least flycatcher might be flying around, the males might be doing different behaviors in the air for the female to look, to be able to determine, do I want to mate with that male? Okay, so they have to do it in the open woodlands. Again, here in the forest, hard to do acrobats and things with too many branches in the way, hard to see one another in the dark forest and with branches in the way. So he might be using calling. So again, it's about the habitat that's important. Okay, so they're both flycatchers, but they are considered different species because this least flycatcher isn't likely to fly into the forest and encounter the other flycatchers to mate with them. They're going to hedge their bets, and of course they're adapted to the open woodlands, so that's where more of their mates are, so they're going to stay there. So again, over time, these may have started out as the same species, and for whatever reason, some individuals found success in the open woodlands while other individuals found success in the forest. But again, over time, because they were isolated, there's no gene flow between these two habitats. Over time, that changed the allele frequencies of each of these groups, and now you have two different populations with different allele frequencies, enough to call it a new species. Okay, so that's habitat isolation, a pre-zygotic barrier, preventing these two groups from mating with one another. Okay. Another one is behavioral isolation, right? So in behavioral isolation, you have distinctive courtship behaviors between the mating species, okay? And so they've really evolved different movements and really precise movements usually the females are kind of watching what is going on. And when she's using this behavior as a means to determine if the male is worth mating with. Does he have strong enough genes to pass on to her offspring, right? Birds are excellent organisms to show behavioral isolation. And so there are a lot of different types of bowerbirds, and bowerbirds are known for making a mating structure. It's not a nest. They don't even mate in this structure. It's just used to attract a female. So what he does is he puts these sticks together, and this species, the satin bowerbird, actually collects items from the forest, but in today's world, we've polluted the area, and so he's actually adapted to collect. For some reason, these birds, maybe females were attracted to our trash. That's more attracted to them, whatever they're cueing in on, the color, the shininess, something. So the males go and collect those now. So a female watches him, he'll do a little dance, and if she likes it, then they'll go off and mate. If she doesn't like it, she'll move on to another bird. Right? So again, you can click, I'll put these on Canvas as well, so you don't have to go to the PowerPoint to click, but I'll put these on Canvas, and so you can see some of the different behaviors that develop. Here's a bird of paradise, also some really cool behaviors. So again, the behaviors become so distinctive, so particular, that it prevents other groups from mating with one another. And then again, you get isolation. You don't get gene flow because now the groups are only picking particular behaviors, and they only recognize that one behavior. Okay. The other one can be, they have to be able to recognize the behavior. Okay. And if you don't recognize it, then you're not going to mate. That prevents gene flow. Okay. Prezygotic barrier. Okay. Next one is mechanical isolation. So in mechanical isolation, this has to do with the actual mating structures. Right? And so it could be structures, the genitalia of animals. It could be the pollinating structures or the flowers in plants, between plants and insects, right? So, so if the genitalia are incompatible with one another, they just physically can't mate. And that's what we see. So here, these are two damselflies mating. This is the male coming around, and he's penetrating the female over here. And they actually have a penis. So this is the male's damselfly's penis. It looks like a scrub brush on the head. And he's actually evolved so that structure fits inside of the female, and he can actually remove the previous male sperm. So a little bit more to guarantee that when he mates with her, that his sperm is going to fertilize her eggs, right? But again, if this evolves in such a way that it no longer fits inside of the female, you're going to have mechanical isolation, right? And it is so specific to this species of damselfly that if he tried to mate with another species of damselfly, it just wouldn't fit. Okay, but you know, again, think of the coevolution between plants and insects, and we looked at this in the beginning of the semester. Remember that plants then develop flower parts to specifically attract certain pollinators, so that that pollinator would only go to that same species of flower, right? And then that way, you're guaranteed the flower is increasing the ability for this pollen to be spread to a flower of the same species. If the pollen goes to a different species, it won't work, right? So if you attract the same pollinator every time and they only like that flower, they're likely to come back. So again, it's the size and the shape of the flower petals and the anthers and the pistils, right, that help to attract, and they'll produce a a smell or a color for that particular individual. And so you can see here under the butterflies with the flower tubes. Here's hibiscus, really specific for hummingbirds and even bats as pollinators. Mechanical isolation. The parts have to fit, right? Or you don't get reproduction. Right? Think about when you see something like, what kind of dog do you have? Oh, that's a cross between a Great Dane and a Dachshund. And we all go, how did that work? Remember, the Dachshund are the little tiny dogs. Great Danes are big dogs, right? You would think there'd be mechanical isolation there. I just made up with those two breeds. I don't know if they actually mate with one another, but Alrighty. The last one considered a pre-zygotic barrier is gametic isolation. Okay, so maybe now the two different individuals, different species, mated with one another. They sperm was delivered to the female, right? But they can't fertilize one another because the gametes are incompatible, right? So the sperm cannot penetrate and get into the egg. Maybe the sperm get lost within the tract because the egg, remember, produces a chemical to attract the sperm, right? Same with external fertilization. Here are sea urchins, here are sea stars. Well, what if they release their egg and sperm at the same time? Okay, well, there's gametic isolation. So the eggs of the sea star release a chemical that the sperm of the sea urchins may not recognize. And then also just molecular differences, that the two cells are just not compatible to join. Okay, so that's gametic isolation, that the egg and the sperm are not compatible. So that's still a pre-zygotic barrier because fertilization was prevented. Okay. For post-zygotic barrier, so this is what we usually associate with hybrids, hybridization. This is when two individuals of different species actually do mate. Okay, but they'll usually have one of the following outcomes. This is much less common in nature, but it does happen. So if two individuals of different species actually end up mating and the gametes fuse, they fertilize, but the zygote may not continue to develop, or it only develops partially in the embryonic stage and it stops. That's called hybrid inviability. Hybrid inviability. Okay, usually the embryo then dies during development. Okay. There are two other types that maybe the individual does, the offspring does develop. Then we might see hybrid sterility or hybrid breakdown. Here's an example of hybrid sterility, right? This does happen. You can have a male donkey. When he mates with a female horse, they create the hybrid, their offspring, a mule, right? So a mule. So in this case, the mule can develop into an adult, but they're sterile. They can't produce themselves. So that again is hybrid sterility. It prevents the interspecific hybrid, so he's considered the interspecific hybrid, that those that do survive to adulthood from reproducing successfully. Okay, and the reason is because the chromosomes numbers are incompatible. Remember when we talked about reproduction, that the chromosomes numbers have to match, right? And this is mostly true in animals. We'll see a little bit something different in plants. But so a horse has 32 chromosomes and a donkey has 31 chromosomes. So when the zygote develops, the embryo develops, it continues to develop so that the offspring survives. But this mule has 63 chromosomes in total, right? So when they when it tries to make gametes, the diploid number cannot divide equally into the two cells. So the cells don't get all of their chromosome pairs. And so therefore they can't create their own offspring, right? And to get a mule, you do need the male donkey crossed with a female horse. If you cross a female donkey with a male horse, you usually get a much smaller animal called a jenny. They refer to it as a jenny. They do exist, but they're not that common because, again, mules can't reproduce on their own. So people do this. People put their donkey and their horse together. Mules are very strong animals, so they're respected on the farm. They're a work animal, right? So they're very strong animals. But when a female donkey and a male horse reproduce and they produce a jenny, it's much smaller, it's weaker, and so you don't see many of those because they're not valuable as work animals. So there's really not a whole lot of reason to breed those, right? So you don't see as many. Okay, it's probably a weaker animal because a female donkey is much smaller than a male horse. And so there's probably limitations in the reproductive system of development inside there. And so it can't develop into a large animal. Alrighty, but a female horse can carry a pretty large animal. All right, that's hybrid sterility. Here's another example. So again, a lot of times these hybrids happen in captivity. They don't seem to happen too much in the wild. It's also hard though to identify it. But here's another example. So in, I think this was Sea Life Aquarium in Hawaii. They put a bottlenose dolphin with a false killer whale. This is called a false killer whale. There's probably a difference of about 5 feet between the two. But they were both single individuals, and these both are social animals, so they decided to put them in the same pool so they could keep each other company. And they were good buddies, but they became more than buddies because they mated with one another and they produced this offspring, right? And this offspring, they called it a wolfen. Okay, but so here's the thing. When you look at this wolfen, Mom, the bottlenose dolphin, has 88 teeth. Dad, the killer whale, false killer whale, has 44 teeth. So the wolfen, if I can get it on here, there it is, has 66 teeth, right in the middle. Right, so, but the wolfen is, I think they thought it was sterile. I think the latest news, though, was that the wolfen did actually reproduce with another dolphin. And so I might have to move this into hybrid breakdown, right? That's the next one. So the offspring of the hybrid might be able to reproduce, but if their offspring then can't reproduce, we call that hybrid breakdown, right? So they end up having abnormalities or something. So there might come a time over time, again, the chromosome numbers just change too much. That over time you won't be able to, the hybrid offspring won't be able to continue to reproduce, right? We see this a lot in plants though, hybrid breakdown. Okay, this is the other third type of post-zygotic barrier. You might have 2 different sunflower species. Only 80% of the offspring are able to reproduce. And so it's just, they begin to, and the next generation, you'll have more breakdown. And after that, so less and less and less are able to reproduce as time goes on. And that's, again, it's the changes in chromosome structure that may cause the gametes with too little or too many genetic material to develop. And then these abnormalities occur, right? Yeah, so that's hybrid breakdown. So post-zygotic barriers, you've got the 3: hybrid inviability, hybrid sterility, and hybrid breakdown. Okay. All right. So that shows us the mechanism for speciation to occur. You need some kind of isolating reproductive barrier. So when there is an isolating reproductive barrier, then how does speciation actually occur? Well, there's 2 mechanisms for it. One is called allopatric speciation and one is sympatric speciation. Right, so again, evolution of species begins with reproductive isolation. You gotta have that first. And then the 2 populations are no longer to interbreed successfully. So that type of reproductive isolation could be temporal, habitat, mechanical, gametic. You can go through all of those. Right, that's the reproductive isolation. Once that occurs, now we have the 2 populations that are no longer to interbreed successfully. But now, how does the speciation occur? We're going to have the gene pools begin to diverge because the allele frequencies are going to change. And so remember, when gene pools are diverging, it's the population that's changing, not individuals. So I keep saying that again and again. I will throughout this unit. Populations evolve, individuals do not, because populations evolve because the allele frequencies change. And allele frequencies are a trait of the population, not an individual. An individual has the allele. The number of alleles is because of the gene pool. Okay? So populations evolve, individuals do not. So again, let's see what are these, how does speciation then actually occur, right? So we've got allopatric speciation or sympatric speciation. So here's allopatric speciation. I think I meant to erase the slide because I have it, you know, I just have it on. I'll go over here. With allopatric speciation, you can get the evolution of a new species from, it comes from an ancestral population, right? And so with allopatric speciation, you've got an ancestral population that gets divided by a geographical isolation from the rest of the species. So the reproductive isolating mechanism here has to be a geographical isolation. Okay, so there's some kind of new selective pressure that's acting on the population. And that's a type of change from the earth. So for example, there used to be a continuity between the Pacific Ocean and the Caribbean, right? The water flowed and so fish could swim back and forth. There was gene flow. But then the Isthmus of Panama arose about 3.5 million years ago. Again, the moving around of the continental plates, they crashed into one another and it rose this piece of land from Panama to South America. That's called the Isthmus of Panama. And so that would separate these two, this population of fish that lived here. So there was an ancestral population that were able to swim back and forth, but then they got separated. All right, so over time, that was a physical isolation, right? That would be physical isolation going on there. Over time, these fish, now they didn't have any more gene flow, so their allele frequencies changed. And so these are two different species of porkfish. And again, you can sequence their DNA and their genes, and there's been enough changes that we call them two different species. When you put them together in the lab, they can't mate with one another anymore either. So that really, again, kind of shows they're two different species. Okay, so again, allopatric speciation occurs when some members of a species become geographically separated from one another. Okay, and this goes hand in hand with what we learned about founder effect and other things like that. So this is allopatric speciation. They have an ancestral population and then experience some kind of geographic isolation. Here's another example of allopatric speciation. Again, it's more likely to occur if the original isolated population is small, right? Because remember, small populations, then genetic drift becomes more prevalent, right? Or more significant. Go back to the early lectures we had on that in population genetics, right? And so that's what we saw. So this is called a pupfish. It's from Death Valley. And its original ancestral population lived in these really large interconnected lakes that were formed in the Ice Age. Okay, so as the ice began to melt, these big lakes were left there. Okay, and these pupfish, there was one population. But then as the climate began to dry, the lakes began to dry, and they began to dry in between one another, and they formed isolated pools. And so again, just by chance, whatever pool you were in when the drying occurred, that's your new population. So this big population in the large lake got separated into small populations in individual lakes. It's a physical geographical barrier. Now they're reproductively isolated, right? And with genetic drift, we have different species of pupfish now. Okay. So each of these, so even you can refer to this habitat isolation because in the different pools, you have different temperature, different oxygen levels, different salt levels, salty. So they've all adapted into these different habitats now. This is a Hawaiian goose called the nene. It was originally from volcanic mountains, and their feet were not completely webbed, right? Their toenails are longer and stronger, and their foot pads are thicker, and they walk on lava flows, right? So, so this is a population of goose that derived from those geese that live down in the ocean. But again, they went up into the, or actually, I should say it in the opposite way, because the population originally were from volcanic mountains. And so then as they started going down toward the water, they started to get more webbing and things, right? So allopatric speciation, being separated by actually a mountain range here from the volcanic mountains. Okay, sympatric speciation is a different one. Okay, with sympatric speciation, you can get different species that are not geographically isolated from one another. So the new species evolves within the same region as the parent species. This is a little bit harder to understand. Okay, we see it more common in plants than animals, and especially in plants. So plants oftentimes don't have geographic barriers. You see different plants, different flowers and things all in the same area. And so you can't control, even though many of the angiosperms developed to co-evolve with specific pollinators. Wind still blows pollination. That pollinator might visit another flower and check it out, right? And so pollen does get on different flowers. Gymnosperms, remember, it's all wind pollination. It all just kind of goes. So, so once in a while, fertilization does occur, right? And that just causes a change in chromosome number. So that's what you see here. Here's species A and species B. You can actually see the diploid number in A is 6, but the diploid number in B is 4. And pollen, they cross-pollinated, right? Here are their gametes. This is the haploid number. And they came together producing a diploid hybrid of 5, right? And so when this divides into gametes, right, actually what happens, there's an unequal distribution, okay? And so they're able to double the chromosome number here. They just end up doubling their chromosome number. So 5 doesn't divide equally into 2 different cells, but 10 does. And so it's just a quirk that happens. So in the division, in the regular mitosis, mitotic division, right, when they double their chromosome number, now you've got a diploid number of 10, and that can produce haploid gametes. But now this gamete has 5 chromosomes, whereas, the parents had 3 or 2, right? So that can work. So this can happen usually through self-fertilization. Remember, some plants can self-fertilize, okay? If it doesn't double, then again, the chromosomes don't pair up, and meiosis just doesn't work correctly, and so you just don't have viable gametes, right? So we saw this is a change in chromosome number that occurs from the parent plants, right? You have 6 and 4, the diploid number here will be 10, okay? So it results in what they call allopolyploidy, okay? A polyploid individual has more than 2 sets of chromosomes. Ploidy is what we refer to as the ploidy number, diploid, haploid. So that's what we refer to. So this is a hybrid, okay? But it formed through sympatric speciation. There was no isolating mechanism, formed in the same environment, right? And again, much more, plants seem to be able to tolerate different ploidy numbers than animals, because it's the spontaneous doubling of of chromosomes has been documented in many plants, but in just very few animals. Okay, but sympatric speciation has been seen to occur in animals, and that usually happens when in the same area there are many microecologies that are available to exploit, right? And so they adapt to these little micro habitats. Okay, and so these fish called cichlids are a good example of that. So cichlids are a fish that live in Africa, in the freshwater lakes in Africa, mainly like one of them is Lake Victoria. It's a real famous lake out in Africa. And there are many different ecological opportunities in the lake. And so some exploit feeding off the bottom, some eat organisms in the water column, some eat organisms that are floating on the water. And there's enough food in each of those areas and they don't deviate from those areas. So over time, what happens is you see that the fish that feed on the bottom, their mouths have actually evolved to turn downward. And they can't eat at the surface anymore because their mouths can't change and grab enough food that way. Okay, so that was a difference enough in speciation. You stay on the floor of the lake, you're not going to mate with the species above you. Okay, that's their habitat, and vice versa, right? Here you can see they're pointing out, even then they evolve different coloration to attract the mates of their own species, right? So different jaw and teeth shape. Yeah, so Lake Victoria, there's 500 endemic species that evolved from one ancestor, maybe a few ancestors. This occurred 12,000 years ago. So that's a sympatric speciation. Okay, so again, remember you gotta make sure you don't confuse the isolating mechanisms with the actual mechanism to get speciation. So speciation occurs either sympatrically or allopatrically. Both of them need to have some kind of reproductive isolating mechanism. And you can see in geographic isolating mechanisms, it is a physical barrier. Okay, in sympatric, this is where you might get more behavioral isolating mechanisms. You might get more temporal isolating mechanisms, things like that. All right, thank you everybody. Hope you enjoyed learning about species.